Here. Recording. Good. All right. And this one is known for two of the components as the RAS MAPK pathway. When it is activated, it stimulates cell division in many cell types. Not all cell types, but many cell types. That's why when things go wrong with it, it can stimulate abnormal cell division, aka cancer. Second thing, it's very important development because it also can stimulate changes in gene expression. Cells can start activating genes that they had before or shut others down. Meaning cells can start differentiating, turning into more specialized cell types. So this pathway is widely used in all kinds of different developmental events. So we can say stimulate cell division and or cell differentiation. Second thing, this pathway is not using any G protein linked receptors. It does use a G protein, but a monomeric G protein known as RAS. So it indirectly activates this G protein known as RAS, which is, we recall when we mentioned about monomeric G protein, we put them in what we call the RAS Superfamily. Because RAS was the first one of these guys discovered and sort of like the granddaddy of the whole clan here. It's very, very important, and it's heavily involved in signaling, and especially in developmental events. Okay, now in this case we're talking about the receptor never touches RAS at all. RAS will not bind to these receptors. Instead, the receptors activate some intermediary proteins which then activate RAS. So we've got a couple steps between receptor and RAS activation. Once RAS is active, then the third thing is our amplification. And the amplification is a series of a three-stage set of kinases. You activate one set of kinases, that activates a second set of kinases, that second set of kinases activates a third set of kinases. So by the time this pathway is activated, you're swimming in protein kinases that do all kinds of different things in cells, especially in gene regulation and stimulating cell division. So we can say activates, we'll say cascade of protein kinases. Okay, those are a couple key points about this very important pathway. Now, these receptors bind to ligands that are small proteins that are known as growth factors. And we got to take a little look at growth factors, see what they are, and a little bit what they do. So let's say receptors bind growth factors. Okay, what the heck is a growth factor? Well, with a name like that, it sounds like something that stimulates cell growth, specifically cell proliferation. Now, what they are small secreted proteins that put out many different cell types. Virtually all cell types will produce and secrete one or more, usually more than one, different growth factors. Okay? Secreted proteins that stimulates cell division and cell differentiation in target cells. Each cell responds to certain growth factors and not to others. Each cell also produces certain growth factors and not others. So a whole bunch of different growth factors around. Now let's do a little historical interlude, a little nostalgia trip in how this came to be. Okay, you go back in like the 1940s, even 1950s and stuff like that. People have been culturing bacteria in petri dishes and stuff like that for, 100 years or more, right? So people start thinking, oh, wouldn't it be nice to grow animal, especially human or mouse cells in culture? So let's try it. So they get their petri dishes and they throw in a pile of stuff that has all kinds of goodies and nutrients and what have you, just like bacterial thing. And they throw these cells in there, and what happens? Not much. The cells kind of cringe up and round up. Their metabolism is very, very slow. They don't seem to divide. They don't behave normally. And eventually the culture dies on you. So people kept on trying to get these things to grow, these human cells and mammalian cells and stuff like that to grow in culture, and they just couldn't do it. And then they figured, okay, well, let's try some cells that are really good at proliferating rapidly, cancer cells. So you get some of these, and even that wouldn't work. And then there was this woman named Henrietta Lacks in Baltimore in the early 1950s, had a very aggressive and nasty form of ovarian cancer that later on killed her a year or so later. But her doctor was also a researcher, decided to give it another shot. So he took some of her ovarian cells. And then he was thinking this. Inside the body, there are all kinds of cells are bathed in blood, lymph, all kinds of body fluids, and maybe there's something in that that just might be necessary for these cells to behave normally. So he got some blood serum from like a horse or something, and threw some of that in the culture media, put these ovarian cancer cells in there, and lo and behold, they started acting normally, at least normal for cancer cells. They start proliferating rapidly, and they were growing like crazy. Unfortunately, they're also growing like crazy in Miss Lax, and it cut her down in not too long a period of time. But he started these cells as the first human cells to be cultured. Epithelial cell, ovarian cancer, and the ovarian epithelium, very aggressive kind of cell. And they called them HeLa cells, for Henrietta Lacks. And they're still probably the most widely used cells in culture right now because they grow real easily. People have done a lot of work with these guys and stuff. It's been pointed out that the total number of HeLa cells in the world would probably equal many, many times the weight of Henrietta Lacks, and she was not a small woman by any stretch of the imagination. There are probably literally tons and trillions of HeLa cells growing in labs all over the world. And they grow very nicely in culture because they came from such aggressively growing precursors and stuff. And think of this, Henrietta Lacks died in 1953. These cells have been growing continuously for the 60 years since they were first isolated in labs all over the world. Okay, so what's the stuff in blood serum? And they also found that fetal organisms, it worked even better. Serum from embryonic organisms worked even better. So there was mysterious things in this blood serum, and he called them growth factors. They stimulate cell growth or proliferation. And the word factor means you got something that does something, you have no idea what the hell it is. Next problem is try to figure out what is a growth factor. What are they? Well, that took many, many years, but eventually it was found out that growth factors are families of small secreted proteins. And they stimulate differentiation and division in the appropriate cell types. There are many different kinds of growth factors. Many of them use the RAS MAPK pathway, but many of them do not. For instance, there's a whole set of growth factors in the immune system and the blood that stimulate differentiation of red blood cells and white blood cells from the precursor cells, things like that. And they're called cytokines or colony stimulating factors. They use a completely different pathway. There are other kinds and stuff as well. We'll look at a couple other growth factor pathways. But many of them do use this particular thing. So let's take a look at a couple of the types of growth factors that do use this pathway. There are many others. Most of these growth factors are not single proteins, they're families of related proteins, each with a specialized function. So a given family may have scores of different members, each having specialized functions. So let's take a look at a couple of them. One called the epidermal growth factor, or EGF family. It's not just differentiation of the outer layers of the skin, it does play a role in that, but many other kinds of things, especially various kinds of what we call mesodermal tissue, stuff like connective tissue, things like that. Whole bunch of EGFs and stuff. Certain overactive EGF receptors are involved in certain kinds of cancers. There are drugs now that will inhibit those receptors. And if anybody's ever heard of a drug called Herceptin, Herceptin inhibits certain kinds of EGF receptors. It has been used clinically for several years now. Okay, that's one family. Another big diverse family. The fibroblast growth factors, or FGS family. Fibroblasts are the cells that produce things like cartilage, connective tissue, and stuff like that. They crawl around and they secrete huge amounts of this material that makes up tendons, ligaments, cartilage, whatever the case is. FGS stimulate that process, but the FGS do a whole bunch of other things. They're involved in a lot of things, including things like limb formation and organization. A lot of different so-called mesodermal tissues and organs use FGS of one kind or another. So they're widely used, the fibroblasts, growth factors. Okay, another one. What's sometimes called nerve growth factor, member of a family of proteins called the neurotrophins. These are growth factors that stimulate differentiation of nerve cells from their precursors. Nerve growth factor, or NGF, is necessary to keep mature nerves alive. In the absence of NGF, they will die on you. Now, interesting thing about the nerve growth factors. These have the potential of repairing central nervous system damage, at least partially. Several years ago, some people did an experiment with mice. They took these mice and they severed the spinal cords of a whole bunch of mice just around here or so. And of course, when you do that, mouse is going to be paralyzed in its hind legs and stuff, and its trunk, lower part of the trunk and stuff, so it's not very good for the mouse. Now, control, they did sort of a sham treatment, but experimental ones, they applied NGF and various neurotrophins to the severed spinal cord. And you know what happened? It partially grew back and partially reconnected. The treated mice were able to have limited use of their hind legs. They couldn't run around like normal mice, but they weren't dragging their hind legs uselessly behind them either. In a human equivalent, somebody got a spinal cord injury, it might mean you could maybe use a walker or perhaps even a cane if you were lucky. So there's instead of being completely paralyzed from the waist down. So there's a lot of interest in this. By the way, it was Christopher Reeve, the actor who played Superman, broke his neck in a horse accident. He was pushing very hard for that kind of stuff in stem cell research. And of course, he was prominent enough that he'd have some sway and go speak to Congress and stuff like that. Yeah, so there's some potential of this in repairing central nervous system damage. Still a long way from coming to the doctor's office. But once again, here again, these growth factors can be very, very, very important. Okay, well that's an example of growth factors. Now let's take a look at this pathway. Look at the clock. I better try. Yep, I think we're going to do another segment.